The stage is yours.
Thank you, Max. First of all, thank you for the invitation to be here and to present our work and share our work with you. And I hope that I will show you some examples of the work that we have been doing that were started to be very focused on the developing of nanomaterials to deliver drugs and biologic molecules, but it became very focused on the immunotherapy field, but using always nanotechnology to modulate the function of immune cells more than targeting the tumor cells, for example. Let me just move. Okay, so I like this graph, and usually I start my presentation showing it, which represents, it's very easy. Well, it looks busy, but it's kind of easy, showing that what is the percentage of drugs or potential medicines that are under clinical development from phase 1 to reach the market. So if they passed phase 1, it means that they were already recognized as being safe. So if you look at oncology, only 5% of those that we already knew that they were safe, but they proceed to phase 1, phase 2, where you will really look into efficacy, only 5% of those reach the market. But with the advancement of the molecular biology, sequencing, bioinformatics, machine learning tools, we are learning more and more about the tumor microenvironment. So nowadays, the tumor cells are not seen as a single entity. And if we manage to characterize the tumor of the patients, we will be able to target our medicines to specific overexpressed proteins, for example, or molecules that are related to the progression of the tumor, even if these molecules are secreted by immune cells, more than tumor cells, for example. So this is driving the development of advanced technologies, such as the CAR T cells, for example, that if you look, the likelihood of approval, it increased. So in oncology from 5 to 17%, or you have the genetic therapies with the delivery of sRNA, for example, that you have 13%. It's not brilliant, but still it's much better. And these numbers are much better, and they reach the 50% when we know and which biomarkers are related to the disease development or to the response to the therapy. And one of the examples that we have is, for example, when it was the development of the trastuzumab, the anti-HER2 antibody, that when the clinical trial was designed, it was designed with a broad inclusion criteria, so the patients would have to have breast cancer. And then when the data was analyzed, you could see that there was no advantage in terms of survival or regression of the tumor. But the principal investigators understood that a big fraction of those patients were expressing actually the HER2, so the target of the antibody. And when they analyzed the data again, but looking at that specific population, then it was a clear advantage in terms of survival and also decrease of the tumor size. So that's why the antibody was approved. So this is just an example that we should be more informed in the way that we are also developing our medicines. And this is what we are trying to do, because in our field, and this is really to the people from pharmaceutical technology to be blamed, is that we were used to develop nanosystems or even microparticles as considering that the composition of these particles would be like excipients in a pill, for example. So they would not have any effect. They would just give you the matrix to enable you to deliver the biological molecules. But in fact, we are learning that this is not the case. And it is important not only due to the size. So when you go from micro to nano, the way that these molecules behave are totally different, not only in terms of the accumulation, but also the way that it changes the function of these cells. So the size is important, the charge is important, but it's also important the composition. And I hope that today I will show you some examples of these. So in our group, we started focus on the immune component related to the tumor. This is a cartoon that I like a lot. It was drawn by Hanahan from the École Polytechnique in Lausanne. And he updated in 2022. I think the first version was published in 2010. And when he elucidated it now more recently, it was to recognize that it's also, for example, microbiome, senescent cells, plasticity of the tumor cells, but also of the immune cells in the tumor microenvironment, and also the immune compartment, that are all contributing for the way that the tumor cells modify their behavior in order to proliferate. But if you look, and most of these mechanisms that are here listed have a common player, which is the immune system. And that's why we are trying to modulate the immune system, but in a way, using that modulation to also overcome some of these tumor-related mechanisms. So the immuno-oncology or the immune-oncology cycle is quite simple. So we have antigen-presenting cells patrolling our body, in the dermis mostly, during our life. And at some point, they will detect cells that present some markers on the surface that are not self. So they are recognized as foreigner bodies. So these cells, in that case, they uptake these antigens, they migrate to the lymph nodes. During this migration, they become activate, which means that they will express co-stimulatory factors that you have here, and they will present these antigens to the T cells. So when they present, they give the information to this T cell to get expanded into activated T cells. And these activated T cells will be able to leave the lymph nodes, migrate and search for the tumor cells wherever they are in the body to then induce their destruction. So this is a huge advantage of the immuno-oncology cycle, because you can use your immune system to destroy these tumor cells, even without knowing where are these tumor cells. So it's quite important in terms of, for example, metastatic situations. So we are all aware, right, of the revolution that was brought by immunotherapy that led to the Nobel to James Allison and Onshu. And this was given due to the PD-1, PD-L1 blockades, and also to the CTLA-4. Most of these two immune checkpoints are negative, which means that when they bind to the ligand, they will block the effector function, the cytotoxic function of these T cells. But you have also some agonists, which means that they are antibodies, that when they bind to the ligand, even if the ligand is on T cells or in the phagocytic cells, they improve the activation of these T cells. But these ligands, none of them are in the market yet. And also the explanation is because it's more difficult to control the activation of the immune system, because you can also lead to autoimmunity, and this needs to be controlled. So in fact, these immune checkpoint antibodies brought a revolution, brought a solution for many patients, namely, for example, metastatic patients and patients with kidney cancer, the stage 4, where they didn't have any other option. And we are talking about patients that would die in a couple of months. And now we have at least 50% of those patients that after 5 years, they are still alive. But still, there are the other 50 in this case, and we are just talking about melanoma and kidney cancer, that will die. And we have many others, for example, in breast cancer, colorectal cancer, that the best response that we get is 10 or 20%, and in specific subpopulations of these cancers. Usually, patients that have the expression of neoantigens, which means that are proteins that are expressed on the tumor cells that were mutated, which means that they are so unique that they become highly immunogenic, so they activate a lot the immune system. And when we look and when we see in the immunology books about explaining this interaction between the PD-1 and PD-L1, this is the cartoon that you see. So mostly the antibodies come and they block the binding of the PD-L1 that is expressed on the cancer cell with the PD-1. But the reality is much more complex. And we know that it's not only these two types of cells that will meet each other. You have all the other cells in the stroma, like the endothelial cells, the fibroblasts, that they, on their own, they differentiate. They are affected by, for example, cytokines that are secreted by the tumor cells, and they become immune suppressive, as they will also secrete immune cytokines or chemokines that will change the function of the T cells, for example. And that's why we are not able to overcome the progression of all the tumors, or more than melanoma kidney cancer, for example. So here is where we think that the nanotechnology can be of can give an advantage, and we are using exactly nanoparticles, mostly polymeric or lipidic, and the choice depends on the physical-chemical properties of the molecules that you want to deliver inside these carriers. And our main goal is to block the interaction between tumor cells or other stroma cells that you have in the tumor microenvironment, or between the tumor cells and the immune cells. And what we try to do, we try to characterize these interactions, and based on the understanding of these interactions, trying to understand which of them have a higher impact on the immune response. So these are the ones that we will select to be targeted by our nanoparticles. So just to give you a small introduction on the nanotechnology and on the nanoparticles. So a nanoparticle is for a soccer ball, as a soccer ball is for the earth. So just for you to have an idea how it is changing in terms of dimension, and this nanoparticle of so small of 15 nanometers, it has a biodistribution totally different, or it allows the drug that is delivered to have a biodistribution totally different from the one that you get when the drug is injected as a solution, for example. And this is an advantage, but it has also to be controlled, as it can be can induce some safety concerns. For example, if by changing this biodistribution, you have a particle full of lipids and it will accumulate in the liver, so you can destroy the liver of that person. So this means that we need to characterize very nicely, not only the physical chemical of these particles, but also to understand where these particles will go and accumulate in the body. So that's why you have several advantages in terms of regulating this biodistribution of the drug, which is very useful. For example, when the liposomes delivering doxorubicin were approved, and they are first-line treatment nowadays for breast cancer, they were approved not because they increased the efficacy of the doxorubicin, but because they allowed to overcome the accumulation of the doxorubicin in the heart. So patients could prolong the treatment, they could actually complete the treatment and not stop due to the cardiac problems that they were developing. So there are a lot of types of nanosystems. These are the polymeric, liposomes. You have some very small like these that are polymer conjugates. So you just have a backbone of a polymer, and conjugated to it, you have a drug or a peptide. And you have dendrimers. There is one approved as to prevent the transmission of HIV. So these dendrimers have conjugated peptide. And as you can see, it looks like a tree. So you can have multiple copies of that peptide, and they are applied as a gel. So it prevents the transmission of the virus. Here, I just wanted to give you some examples of those that are already marketed. And especially this one, Vixeos, it was really a paradigm change in our field, because it was the first one and the only one up to now where you have a carrier with two different drugs inside. And the special about this is that you have a drug that has a very short half-life. So it means that a drug that when it reaches the blood circulation is degraded very fast. And you have another one that has a very long half-life. So, and these, you wanted to take advantage of the simultaneous effect of these two drugs. So if you inject them in the circulation as a free, one will be degraded faster than the other, so you will hardly have both at the same time in circulation. And this liposome allowed it. And it was very challenging in terms of the characterization of the system, because the people had to quantify and show for how long each of these drugs remain in circulation after the administration of the liposome by intravenous administration. And this sounds very easy, but it was not. So they had to develop an analytical methodology able to quantify both of the drugs with very different pharmaceutical properties in order to convince the authorities that you were taking an advantage over having a synergy following the administration of the liposome, which you don't have when you administered both of the drugs separately. So this was a very good, and it is important because it showed that you really can control what you have inside and quantify in terms of not only molarity, but also in terms of the number of molecules that you will have per liposome or per nanoparticle. So we have to characterize fully and extensively the nanosystem, because we need to show that we have reproducibility in terms of the production, but also because we know that any change that you may have, very slight change, if you have that change in a pill, it will not reflect in terms of safety as if you have that small change in a carrier. So that's why you need to characterize the physical chemical properties in terms of loading, the charge, the size, hydrophobicity, etc. And then show that in terms of the biodistribution and accumulation, where do they go, how long they stay in the blood, and in the tissues of the organs where they accumulate. And then also inside the cells as well. So you see, and the way that we do, it has to be very integrated. So you need to explain why you have that effect based on the properties of your particle. And this is what we try to do, because if you know exactly this, you can predict if you use X numbers of ligands on the surface, you will have higher or lower efficacy. And it doesn't mean that if you have more ligands on the surface that you will have higher efficacy, because it can also depend on the access of the target that you have, if it is on the surface of the cell or if it is intracellular one. So this knowledgeable will increase a lot the number of these drugs that will reach the market. So in our case, we started from very basic steps. So our first idea was to look at the mechanisms why tumors at the time were known to evade the immune system. And one of the major was to hide the antigens from the surface of the tumor cell. So your dendritic cells that were patrolling the periphery of your body would not have enough antigens exposed to become active. So what we thought, it was, okay, so we can use a carrier, and you load this carrier with these antigens, and you give these antigens directly to the dendritic cell, so they will become activated for sure. So in this case, you want to activate these cells against tumors that already exist in the system. So it's a kind of therapeutic vaccine. So what we did, and it was known at the time, that you need to have a balanced immunity. At this time, we were highly focused on T-cell activation, so cellular immunity. So we knew that we must have a peptide that would bind to the MHC2 molecule. And for this, you would have a particle that would be internalized by the endolysosomal pathway, which is the most common, get degraded, release the antigen, and here the antigen can be a protein that will be then processed into peptide segments, or can be directly the peptide. In our case, we use the protein in those assays that we want to learn more from the immunology point of view, because you have very nice immunology models, mouse models and cell models that bind to ovalbumin peptides, ovalbumin as a model protein. But we use in most of the other studies directly in cancer models, the peptides. So we have to have a peptide that will bind to the MHC2 molecule. And this was known already to be a peptide with between 14, 16 amino acids. Can be until 20, but most common is 13, 15 amino acids. And this activation would this presentation of this antigen would activate the CD4 T cells, which are the called T helper, because they help the CD8 T cells to become activated. So you really need to have the activation of these ones in order to have activation of the CD8. But to have the presentation of these peptides to the CD8, you have to have a particle that will not be degraded into the endolysosomal pathway, but will be translocated to the cytoplasm, there be degraded, release the peptide, and this peptide would bind to the MHC1 molecule on the endoplasmic reticulum, and then the complex will be translocated to the surface and presented to the CD8. Then you would get the cytotoxic immunity. So MHC peptide presentation, but you also need to have upregulation of activation markers on the dendritic cells, and the secretion of the cytokines. If you manage to have these three stimulus, you have the right activation of the T cells. And this is what we wanted to do. So I will not present you the earlier works that we did, but just to make a summary that we understood that we have to have these peptides together with adjuvants. So those cytokine secretion and upregulation of the markers we only have if you have inside the particle the antigens, but also the adjuvants. And here adjuvants, you can use several depending on the nature of the immune response that you want. In our case, as we are looking at cancer, and we were more interested in having cytotoxic immunity, but also to have the Th helper activation, we use ligands from the tool-like receptors that are expressed here in the membranes of the endolysosomes or in the reticulum as well. So we saw first that you need to have them together in the same particle. And then we also learned that depending on the type of antigen, you can have that short or long peptides in the same carrier or in distinct carriers. And this is really dependent on the antigen itself. We learned that it was not dependent on the type of tumor, and actually it was not dependent on the properties of the carrier, but rather related to the peptide itself. We assume that is related to the intrinsic immunogenicity of these peptides. And so I will show you three examples of three different carriers, and I will explain to you how we evolved and why we selected the composition and the peptides and the activators as well. So the first example that I will give you, it's against melanoma. And this was a particle that had monos on the surface. The second one, it is a vaccine against colorectal cancer. In this case, we were interested to develop the vaccine where we would avoid polyethylene glycol, because this is a polymer widely used, for example, in the production of antibody drug conjugates, where you have the protein connected to a drug, and this connection is made by a polyethylene glycol. And the reason was because there are already data showing that the patients at some point develop antibodies anti-PEG, which means that these antibodies will destroy the antibodies or the carriers that are with this PEG on the surface. And if you get them destroyed, you will not get efficacy. So we try to explore alternative polymers that would confer the particle the same hydrophilicity. And this hydrophilicity is important so you don't get destruction of your particles before they reach the dendritic cells. And the last example, it was a breast cancer vaccine, where I will show you that depending on the composition of the particle with no target, it also changes the efficacy of your system. So in the first one, as I told you, we already knew that we had, for this case, for the MART1 peptide, we had to have the peptide together with the adjuvant. So in this case, we used the MPLA and the CpG. And we also knew in this case that we had to have the peptides separately. So we had one particle with a short peptide and the adjuvants, and another particle with a longer peptide with the adjuvants. And we also knew that when we gave this vaccine to the animals, we managed to infiltrate a lot of T cells, but at some point they became exhausted. So that's why we thought that by combining this particle with the immune checkpoint inhibitor, we would somehow make the non-responders to anti-PD-1 become responders. And because we knew that we were able to activate these T cells at a certain stage, we also combined with anti-OX40 because we wanted to show, and this OX40, what it does, it's an agonist on the T cells, and it overcomes the proliferation of the T cells into regulatory T cells. So this is what we thought. So we produced these particles without monos on the surface and with monos on the surface. The monos is expressed not only by dendritic cells, it also is expressed by macrophages, B cells, and endothelial cells. So we wanted it to be mostly taken up by dendritic cells. So we played with the decoration of these particles with monos, but also with the size. And we know from the past that if you have bigger particles, like let's say 500 nanometers, these ones are mostly recognized by macrophages. But you have particles that are smaller, like around 200 nanometers, 100 nanometers, they are not taken up mostly by these macrophages, but they will be recognized by the dendritic cells. So this is what we did. We targeted nanoparticles with 200 nanometers, the most, and this indication of the polydispersity index tells you that the size of these nanoparticles was very similar. And you can see here in the electronic microscopy pictures that in fact they were quite similar. And here it's just a histogram showing you that the mean average size, it was in fact around these 200 nanometers, 180. But we wanted to show that this mannose was on the surface. So you know that the concanavalin A binds to 4 mannose molecules. So if this concanavalin A would bind to mannose, you would not measure the average size of one particle, but you would measure the average size of aggregates of these particles. And that's why you see this shift. So the average size shifted to more than 1 micro. So you are actually measuring the size of the aggregate. So this means that the mannose was on the surface. So we injected these particles in the animals. After we checked that the mannose was making a difference in cells, and we could see that they were accumulated in the inguinal lymph nodes. In here, we used sub-Q administration. But when we took the organs, we could see that they were mostly accumulated in the lymph nodes, which was a good news. But we didn't see significant difference between those that had mannose on the surface or didn't have. So we proceeded to a very simple animal setting where we just wanted to learn how this vaccine was activating the T cells. So we first gave the vaccine to the animals, and only after we inoculated the melanoma cells, and we then started the combination with the immune checkpoint inhibitors. And what you could see, it is that the red line, which is the one given by the combination of the particles with mannose on the surface combined with immune checkpoint inhibitors, prolonged or was able to delay the development of the tumor, much more than when you had the particles without the mannose on the surface. And this was translated in terms of survival. So we moved to the therapeutic setting that we were interested. So in this case, first we established the tumor, and we only started the vaccination when the animals had already the tumor. And in this case, we were very surprised and not very happy, because what you could see is it was that the response of the animals that were treated with a monosylated nanovaccine combined with immune checkpoints was very similar to the one presented by the animals that were treated only with the immune checkpoint inhibitors. So this was not expectable. So all the advantage that we saw before here was not being translated. So we repeated the experiment. We took the tumors and we characterized the immune cell subtypes that were infiltrating that tumor at 2 time points. In a very early time point when we knew that the response was very strong, and at a later time point at day 38 when we knew that was already escaped, so the tumor was exploding. So what you could see is that at an early time point, these 2 groups that were the best ones, they were infiltrated by high frequency of the CD8 T cells, which are then leading to the cytotoxic T cells. The CD4 were actually a bit decreased, but this was not relevant and was not a signal of alarm. But at the later time point, the CD8 T cells almost disappeared in the animals that were vaccinated. There was no change in the CD4 T cells, but we saw this huge infiltration. So most of the immune cells that we were having inside the tumor were these cells. And these cells are myeloid-derived suppressor cells. They are highly suppressive. They are reported in the literature. And at the time, we also saw that ibrutinib, which is a drug that is approved for multiple myeloma, and it was approved at the time, was shown to be able to deplete, to remove these myeloid-derived suppressor cells from solid tumors. So to test the hypothesis that these cells were actually those that were responsible for blocking our effect, we tested again. We repeated the same experiment, but now with ibrutinib. And what you can see is that when ibrutinib was added to the dual therapy that we tested before, the delay on tumor growth was really prolonged, much more than when the ibrutinib was added to the immune checkpoint inhibitors. And this was translated in terms of survival. And very interesting, it was also related in terms of a low variability in terms of tumor volume presented by the animals. And this is a very good indication from our experience. When you have a drug that really works, all the animals or most of the animals behave very similarly in terms of tumor growth. And what you have here, this dispersion of tumor volumes that we got with the immune checkpoint combined with the ibrutinib, it's quite typical from what you can see in the clinic with the responders and non-responders. So we introduced ibrutinib to the dual therapy with the monosylated nanoparticles, but at this point we thought maybe this will allow you to have the vaccine without the monos on the surface, and this is a much simpler system as you don't need to work with conjugation before the formulation. So we repeated the same experiment, and in black you have the response of the animals where the treatment was the same, so the immune checkpoints combined with ibrutinib and the vaccine without monos, and in red you have the same combinational treatment but with the monosylated vaccine, and you can see that the monos was doing the work. So it is exactly the presence of the monos on the surface that is changing the way that the antigens are being presented by the dendritic cells. So you see this exactly same carrier. The only difference is the monos. The physical chemical properties are very similar. So it was the monos. Again, we had a very low variability in terms of tumor volume. And these spheres on the right, what they show you is each purple dot point. It's the representing the secretion of interferon gamma by the T cells activated. And this is a very good indication of response. And what you can see is that, of course, it's much, much more darker in those animals that were treated with a triple combination, but with the monos on the surface. So this study was very interesting to us, and it was a surprise because we were not expecting that the monos would make such a huge difference. And we already depleted, for example, the macrophages, and you get the same response, which means that it is due to the uptake of these particles by the dendritic cells, and this is why it is changing the way that these dendritic cells are presenting the antibodies, the peptides. So the story on the colorectal cancer, it was different. We know, and it is quite well known, that in the metastatic settings, you have a lot of cells that contribute to the malignancy of these tumors, but one that is present or having a major role are the macrophages, but also this cytokine, the TGF-beta. This cytokine is mostly secreted by dendritic cells and tumor cells. And in our case, when you are trying to develop a vaccine, it's very crucial because it inhibits the dendritic cell activation, which is our major target of the vaccine. It also inhibits the macrophages, the pro-tumor and the pro-inflammatory macrophages, and it also blocks the proliferation of the CD8 that you aim to upregulate with your activation of the dendritic cells, and at the same time it activates the regulatory T cells that are immune suppressive. So we thought that this would be a very interesting target for us, especially because it's highly secreted by dendritic cells. So Anna from my lab, what she tried, it was to have a particle that would have inside an siRNA able to silence the TGF-beta secretion by the dendritic cells and have as well a neoantigen. In this case, we used the neoantigen of MC38, which is a cell line that was used, for example, for the approval of the immune checkpoint inhibitors. This is a colorectal cancer cell line. It is immunogenic, so it is quite easy actually that it responds to the activation. So then we tried as well other models just to show that the effect was actually strong. And also Anna wanted to overcome this proliferation of the pro-tumor macrophages. So in this case, we wanted to combine the particle with an inhibitor of this receptor, CSF1R, that was being shown to be effective, but not enough. So we thought if we play on the activation of the dendritic cells, while we overcome the secretion of the TGF-beta and combine with this inhibitor that is called pexidartinib, maybe we would be able to have a strong effect. And also, Anna was also thinking, is it enough if I block the secretion of the TGF-beta by the dendritic cells, or I also need to block the secretion of this TGF-beta by the tumor cells. So she developed another type of particle, and I'll show you here, where in addition to the siRNA, silencing the TGF-beta, she had on the surface an RGD peptide conjugated to the polyoxazoline. So this RGD will bind to the tumor vasculature, actually, and this was proved before. So we had these three systems: a vaccine just with the peptides and the adjuvant, a vaccine with the peptides, the adjuvant, and also the siRNA, and the same particle, but instead of having the monos on the surface, we had a ligand for the tumor microenvironment. And you can see that the changes on the ligand and the incorporation of the siRNA didn't change actually the mean average size of these particles, which is good. And we wanted, as I said, to test the difference between the pegylated and polyoxazoline-based nanoparticle. So we established a model in a colorectal cancer model, and then we gave the vaccine. And what you can see is that, in fact, there was a difference, and we were quite surprised that the difference was so strong between the particles that had the PEG on the surface or those that had the polyoxazoline. We took again the tumors, and I'm just presenting you the data on the T cells. And we saw that both particles were successfully infiltrating T cells, but those that had the polyoxazoline somehow managed to get the cytotoxic T cells at a higher levels inside the tumor. So we thought that somehow these CD8 T cells were driving the response on the polyoxazoline nanoparticles, and the effect of the pegylated nanoparticles was not so strong because we didn't manage to activate these CD8 T cells. So to prove this, we repeat the same experiment, and we depleted these CD8 T cells from the mice. And what you can see is that here in orange, you have the effect of the vaccine of the polyoxylated nanoparticles, which was very similar as before. But when you remove these CD8 T cells, this response disappeared. And the effect was also lower in terms of anti-tumor volume when you block the CD8 T cells on the pegylated on the animals treated with the pegylated nanoparticles, but it was not that big, the difference. So this made it very clear, if you compare these two, that it was the CD8 T cells that were mediating the immune response in these animals, which was very interesting. So from here, we continued only with the polyoxazoline-based nanoparticles. So as I told you, we wanted to see if having only the vaccine or the vaccine with the siRNA inside would make a difference. And in here, you can see that it seemed that it did not make a difference. But when we combined the vaccine with the pexidartinib, then the effect was much stronger. That is this one in blue. But when we added as well the particles that were targeting the tumor microenvironment to deplete the secretion of the TGF-beta, the effect was not improved. So actually, the best system was this one that we had the vaccine silencing the TGF-beta on the dendritic cells combined with the pexidartinib. And this was translated as well in terms of the CD8 T cells, but these cells also presented PD-1, which is a marker of activation at an early time point, but a marker of immune suppression at a later time point. And this is what we thought that we could combine the vaccine with the pexidartinib with the anti-PD-1. And this would allow also the patients, in this case our mice patients, that didn't reply to the anti-PD-1 to become responsive. And this is what you have. When we have the particles delivering the siRNA, the peptides and the adjuvants to the dendritic cells, combined with the pexidartinib and the anti-PD-1, the effect was much stronger than when you had all these components free in solution. So the particle makes a difference. Again, if you see the tumor size presented by these animals, it was very similar between all of them compared to those that were treated with the free component. So the carrier was making a difference. But as I told you, this MC38 is quite immunogenic, and it is quite easy to get a response. So we tested the same system, but now against a CT26 colorectal cancer model. It's also immunogenic, but in this case, we didn't use a neoantigen, and the neoantigens are much stronger. In this case, we used the KRAS antigen that is also present in a high percentage of colorectal cancer patients. So, and what you could see was that the effect was the same. And in this case, we also evaluated if the combination of the pexidartinib with anti-PD-1 would be enough, because also this model is immunogenic, so maybe you would not need the vaccine. But we have shown that actually you need the vaccine to have this effect in terms of control of tumor growth and also in terms of survival. And none of in the treatments we saw a huge variability in terms of body weight of the animals. So it means that the vaccine is being tolerated by the animals. So we moved on and tested the same system in a non-immunogenic model, actually a model that doesn't reply to immunotherapy, which is a melanoma model based on B16F10 cell lines. In this case, we used the neoantigen, and what you could see, the effect was very similar. So this means that the effect that we were having by modulating the secretion of the TGF-beta by the dendritic cells and the blockage of this proliferation of the tumor, of the macrophages into tumor macrophages was being attributed to the carrier itself. So it is not related to the cell line that we were using, as we tested in 3 different cell lines, and these cell lines actually have different induced tumors with different immunogenic profile, and in the 3 cases you had efficacy. So in the last, let me just see the time, I promise I will move faster. In the last case, it was in breast cancer. Also in this case, we have a problem. Only around 20% of the patients respond to the therapy, but in this case, most of these patients, after a couple of years, they regress. And it's especially worse in the cases where you have triple negative breast cancer, where you hardly have markers, hardly have antigens. So we thought that this could be a nice model to challenge the system. In this case, I can tell you, I will not show you, but we did the same experiment with the monosyllated nanovaccine. And against this tumor, the monos didn't have any effect, so any advantage. So we had to think about another system that would allow us to overcome the immune suppression that we were seeing that was mostly related to regulatory T cells. So what we thought it was to develop a particle, also polymeric one, but in this case to have hyaluronic acid, not because it is a ligand of the CD44 receptor on tumor cells, but because it was started to being, it's not still clear, but it is definitely an adjuvant. And at the time, it was being claimed that its effect was more on the T cell activation. So that's why we used it. At the same time, we used the sRNA depleting the TGF-beta that we saw that the particle was able to do. And also we used the pluronic as a surfactant that was milder in terms of formulation. It was poorly in terms of development, formulation preparation. And to deliver the sRNA, in this case, we tested also a polymer that is based on chitosan, which was also revealed to be an adjuvant of dendritic cells, but we tested different kinds of chitosans because it was reported in the literature that depending on the type of chitosan, actually the suitability or the ability of the sRNA to deplete or to silence the secretion of the cytokines would be different. And we saw this, that when we use the glutamate chitosan compared with the hydrochloride chitosan to complex with the sRNA, the silencing of the TGF-beta was different in terms of the mRNA levels. We didn't see difference in terms of the protein levels, but maybe this is just a matter of time point. So, and we saw both in tumor cells and also in dendritic cells. So we moved on. The antigen that we used, and this is a model of triple negative breast cancer, it was lactalbumin. Lactalbumin, it's an antigen that is present only in 2 situations, during pregnancy and lactation, which means that you would not be able to give this vaccine to pregnant women or women that are lactating. But still, it is a quite interesting antigen, especially in a type of tumor that you don't have many options. So we also combined with the anti-OX40 because, as I told you, we saw that we had a huge presence of regulatory T cells. And what we could see, it was actually quite surprising, because when we gave the vaccine or the OX40 alone, we didn't see any effect. And you can see here in terms of tumor volume. But when we gave both together, the tumors kind of became dormant, and we only started the treatment here when you have quite big tumors. So you know that you have the tumors. So we took these tumors. We also checked if you had decrease on the secretion of the TGF-beta, so all was good. And we took the tumors and we saw that only this group presented a huge level of CD8 T cells. So those T cells, CD8, were activated. And this is given by your CD107 marker. Very interestingly, we saw a upregulation of the 3 cytokines that are like the gold activation markers, the interferon gamma, IL-2, and TNF-alpha. We saw a huge infiltration of CD4, but the regulatory T cells were not different among all the groups. So this was good. So it means that these CD4 T cells that were infiltrating, they were actually effector T cells. So again, to show that the effect is not related to the cell line that we are using, we did the same experiment, but now we use the luminal B breast cancer that is also affecting 30% of the breast cancer patients, especially those that are quite young, so below 40 years old. And again, we saw the same effect. In the nanoparticle alone, we didn't see a huge effect. The 3 components combined with OX40, it was a kind of efficacy, and you can see here some responders and some non-responders. But when we combined both, the effect was much, much stronger. And again, the variability in terms of tumor volume was very low, and the survival was very impressive. So I will not, it's quite complex, but I will just tell you that we saw that again, the infiltration of CD4 and CD8 T cells were confirmed, the decrease of the regulatory T cells. And very interestingly, we saw infiltration of NK cells, which means that at this point we were able to modulate the adaptive immunity that is given to you by the CD4 and CD8 T cells, very simple. And the innate immunity was also working. And maybe that's why the previous vaccine didn't work in these settings, because with the monosylated nanoparticles, we were mostly working on the adaptive immunity. We didn't have a huge change in the NK compartment. And in this case we had, and we saw that this effect was actually due to the presence, and this was a huge surprise to us, to the hyaluronic acid we were expecting actually, but not that the presence of the surfactant that we simply selected because it's milder than the polyvinyl alcohol, but only when the 2 were present, which is given by your red column, the effect was much stronger. So what you can see here is that we tested the nanoparticle produced just with the polyvinyl alcohol as a surfactant, so without the pluronic and without hyaluronic acid. In this bright blue, you have the polyvinyl alcohol with hyaluronic acid. In this one, just the pluronic, and in this one with both. And it was clear that the composition of the particle, it makes a huge difference. And now we are trying to understand, because this, for example, the pluronic, it is expected to be washed from the particles after the production, and what you get there is around 2%. But this 2% is making a difference, and we had to have the presence of these 2 components to have the innate immunity effective. Just to show you some mechanistic studies that we are doing, just to try to understand why these particles are working. And with a collaborator that is a immunologist, because he was very skeptical about the carriers. He was not believing how the matrix itself, and by simply having the antigens and the adjuvants inside the particles, were changing and giving you this activation. So he repeated the experiment, now using the CpG and ovalbumin as an adjuvant. He took the blood of the animals, and he saw a huge secretion of IgG2A, which is an antibody indicating cytotoxic immunity. But then he also took the lymph nodes, and he saw that in the lymph nodes you have this dot, this huge amount of cells here, which means that you are having activation of the B cells. And these B cells were able to activate these T follicular helper cells that are a type of T cells inside the lymph node that will give you a huge activation of humoral immunity. That's why, and this explained why you get a huge secretion of these IgG antibodies. So it took the lymph nodes, it sorted the T cells, these T follicular helper cells, and what you have in yellow and in purple, in yellow are the T cell receptor genetic profile of these T cells that were collected from animals treated with an antigen and the adjuvant free in solution. And in purple, you have the same T cell receptor genetic profile, but presented by the T follicular helper cells that you sorted from the lymph nodes. And you can see that the genetic profile is totally different. And now we are trying to understand what does this mean, which change in the profile of this T cell receptor is inducing and correlated with the efficacy, because then this can also be a knowledge that can be applied, for example, to produce CAR T cells more efficacious. So in some situations that you can get more higher efficacy, for example, against lymphoma or some leukemias as well. So I hope I showed you that these carriers actually, they are very versatile. We must understand better what is the impact of changing the composition of these systems on the biological response of the host, but they are quite flexible and we can play with it to have more humoral immunity, so antibody secretion, or CD8 or CD4 T cells, or even innate immunity, depending on the tumor that you are treating. And it can also be used for different administration routes. We are using, for example, for nasal administration when you have metastases in the brain. So to modulate outside of the brain the immune response, but then the immune cells infiltrate and attack the metastases inside the brain. And of course, this is done with a huge network of collaborators that I thank you them all, also the funders and all of you for your attention. And Max, again, for the invitation.
Yeah, so thank you very much for the talk, Helena. My question is more like coming from a technical point of view, is like how important is simulation for the things you're doing? Because I think from what you told us, it mostly sounds like that you're preparing the nanoparticles, thinking about what could work, then you do testing, then you do animal experiments and so on and so on. So how important is currently their simulation, for example?
Yeah, I think it's key, to be honest, because what we are doing, and when I say that we need to understand better how even the composition of the particles or the decoration of the particles will impact the interaction of these particles, for example, with the dendritic cells, but also on how these dendritic cells then will interact with the other immune cells, we have to do, as I explained to you, all this work. So we need to prepare the particles with and without, and then test it in cells and then test in animals. And I think with simulations, you can overcome a lot of these steps. Because, for example, especially with the mannose receptor, we know that the mannose will interact with the mannose receptor. We know where they are and when they are exposed. So I guess with simulating the presence of these mannose, and also with the difference of the sizes of the particles, and how they will interact with the target cells, will help us also to have a kind of design space where we can play with the production, so it will be much more informed. And also then when you have the particles and see how they will interact, for example, with the cells in a culture. For example, we have a lot of debates of how many particles we should incubate with the cells. Does it make a difference? What is the limit? So I think, and this is a field where the simulation is starting to be very important and integrating with the production and development of the particles, definitely.
Yeah, that's promising for us, I would say.
Exactly. No, it is actually. I'm getting some proposals where exactly people are starting to look. Of course, it's easier if you start to do it with lipid nanoparticles, because the lipid nanoparticles are more, let's say, known. So there are a lot of knowledge. For example, if you change to this certain type of lipid, they will go to the liver. If you change to this lipid, it will go to the bone marrow. So I think you have more information there as a starting point. But I think that's why the polymeric nanoparticles represent a huge opportunity, because it's also nothing is there done. So everything and all the knowledge that we may acquire, it can help. Of course, we will have to get more information to also give you the tools to build these simulations, right? But I think it will explode this field for sure.